Welcome back to my course Aspects of Biochemical Engineering. In last couple of lectures, we tried to cover the theoretical part, then analytical part of that microbial system, how the kinetics of substrate utilization, product formation, and biomass formation by using different microorganisms can be explained and or can be analyzed. So we started with that how we can monitor the cell mass concentration. Then we discussed what are the different models we have. We discussed the structured model, unstructured model, segregated model, and unsegregated model. Then we discussed about the inoculation of the microorganism using the life cycle. Then we analyze the batch process, we analyze the continuous process. As per continuous process is concerned, there are two type of process. One is chemostat or CSTR, another is plug flow reactor. And another process we explained, that is the fed batch process, which is mostly used for where the substrate acts as an inhibitor. Now after that we discussed different type of models. We discussed the chemostat process in details. The major drawbacks of the chemostat process is the cell mass wasting from the reactor. If it is more as compared to the cell mass that is growing in the reactor, obviously that a time will come when there will be no cell present in the reactor. The situation what we call de-washout that will arise. So we try to discuss what are the avenue through which we can safeguard this particular process. One approach is cell mass recycling, another is the immobilization of the whole cell. So we discuss both the process in details, that CSTR with cell mass recycling and also whole cell immobilization system, how it is used in how those processes used in the industry or different biochemical processes. Now today our purpose is little bit different. We want to discuss the couple of lectures will be concentrated on the different numerical problems, and I personally feel that more we solve the numerical problems, your idea on this particular biochemical process will be clear. So with this objective, the first problem that we have taken into consideration, that is the batch process, because that when cell grow in a particular system, that question comes how you can monitor the kinetic constant, as for example maximum specific growth rate of the cell and the saturation constant. Now this problem is little bit dealt with this. You see that Escherichia coli is being used for the production of recombinant this porcelain cell growth hormone. The bacteria are grown aerobically in a batch culture with glucose as the growth limiting substrate, and cell and substrate concentration are measured the function of culture time and results are listed value. So we understand what is called batch process. Batch process, I told you, we take the material at a time, we take the media, we take the medium and we put the cell here. So then we put all the things together, and then time to time we draw the sample here, we draw the sample here, and we monitor with respect to time what is the cell mass concentration, what is the substrate concentration. So this is exactly this problem deals with like this, and these are the different their experimental results that has been given. Then what is finally what they are asking for, that what is the value of mu max and Ks, what you call maximum specific growth rate and the saturation constant Ks, and what is the observed biomass yield of the substrate. That is these are the this is the parameters we shall have to monitor. So we know that to solve this problem we shall have to take the Monod equation. Now we know the Monod equation. What is the Monod equation? Monod equation is mu max, mu equal to mu max S, Ks plus S. Am I right? Now, mu, what is mu? Mu is the specific growth rate. This is called specific growth rate. This is the specific growth rate. And what is this? S is called the limiting substrate. So, once what we shall have to do to solve this particular to find out the values of mu max and Ks, what we shall have to do, we shall have to put the line, write the line over buck plot using the Monod equation, and this will be what? Ks, this is Ks by mu max 1 by S plus 1 by mu max. Am I right? So, if you plot now 1 by mu versus 1 by S, we will get a straight line. The slope will give you the value of, this is Ks by mu max, and this intercept will give you the value of 1 by mu max. Am I right? So, this is how we can find out. So, this mu max value you can put it here, we can find out the value of Ks. Now, question come, how you can find out the value of mu. So, in this problem what you have, we have T, we have X, and we have S value. So, different value in this problem is given. Now, what we shall have to find out? We shall have to find out what is dx by dt. Now, I told you the dx by dt, how we can find out? This is equal to x in plus 1 minus x in minus 1 divided by 2 del t. Now, here n is the sampling number. This is sampling number 1, 2, 3, 4, 5, 6 like this. So, simple. So, by using this equation we can find out dx by dt. Once we know dx by dt, we know the at this point what is the value of x, and at this point we know the what is the value of dx by dt. So, we can find out 1 by x, value of 1 by x dx by dt. This is nothing but mu. Am I right? And once you know the mu value, then we can find out the value of 1 by mu, and simultaneously we can find out 1 by S value. S value is given here. So, we can plot 1 by mu versus 1 by S, and we can get the kinetic constant Ks, and this we can easily find out. Now, this is the table that you can see that and different values are given. This is the x value, 1 by x value, 1 by s value, dx by dt. This is how we can find out, and then we find out the mu value, which is nothing but 1 by x dx by dt. Then 1 by mu value we can find out. Then we can plot. This is 1 by mu versus 1 by s plot. Then we will get a straight line, and from the intercept we can get the value of 1 by mu max, that is coming 1.69 our inverse, and the slope is 0.65. You put the value of mu max here, then we will get the K's value 1.1 kg per meter cubed. So, we can find out the kinetic constant. Now, question comes that next is the what is the observed biomass yield, the ratio. So, what we can write that y x by s is equal to dx by ds. Am I right? And this we can write, this is equal to del s del x by del s. This is equal to x minus x 0 by s 0 minus s. So, we can from this ratio we can find out that from the table x we can taking the value at time 3.7 hours, we get the y x by s value 0.46 kg per kg. So, we can easily find out the values of the different kinetic constant of any microbial system. I hope everybody can do it very easily. The next problem that is very interesting, that is also with respect to the batch process, and this problem is interesting. The reason is that because when you run a particular batch process, we are interested to know that after a certain time what is the cell mass concentration, what is the substrate conversion. So, how you can monitor, how we can determine this. This problem will deal with that. Let us see how we can solve it. Now, here a strain of E. coli has been genetically engineered to produce human proteins. You know that one human protein is the insulin. That is this is the recombinant protein that we produce through the microbial fermentation process. And we and this a batch culture is started by inoculating 12 gram of cell into 100 liter fermenter containing 10 grams per liter of glucose. The maximum specific growth rate of the culture is 0.9 hour inverse. Biomass yield from glucose is 0.575 gram per gram. So, first problem that we have, estimate the time required to reach the stationary phase, and what will be the final cell density if the fermentation is stopped after 70% substrate is consumed. So, this is very typical. This is a common problem that we have with the batch process, that we are interested to know that how much time is required for the attaining the stationary phase. I can explain like this. If you know the life cycle of the cell. So, what is the life cycle of the cell? This is X is the cell mass, viable cells, and this is time. So, we will be having this. So, this is lag phase, this is log phase, this is stationary phase, and this is death phase. So, the question is that we are in this problem, what is the time required to reach the stationary phase. So, we shall have to find out this value T1. Now, question comes how we can do that, how you can find out that, because that we shall have to find out. Now, we know that what do you know, that mu equal to 1 by x dx by dt. Am I right? So, dx by x I can write mu into dt. This is dt, okay, and this is d ln x mu into dt. Am I right? Now, this is equal to x 0 by x, and this is 0 to t. Now, one thing here we can assume, they are saying that after the end of log phase, that what is the cell mass concentration, then stationary phase starts. We can assume this mu equal to mu max. The maximum growth rate of the cell occur in the log phase. So, if you assume that, then our situation is very simple. We can write this is the T batch process. Now, T batch will be what? T batch will be equal to 1 by mu max and ln x by x 0. Am I right? So, this is exactly what we have done in this problem. So, this is the equation that we have written here. This is ln Tb equal to ln x by x 0 by mu. Now, once you have this, we know that this is the yield coefficient is y x by s equal to x by x minus x 0, s 0 minus x. The x will be equal to what? This y x by s s 0 minus s. So, what was our previous equation? Tb equal to ln x by x 0 divided by mu max. Am I right? So, here 1 by mu max I can take it out. Now, x will be equal to this. So, I can put this value here, and this is the x 0. So, we can simplify in this form, and then we know we assume that at the stationary phase there is what is the stationary phase, how we define stationary phase? We define stationary phase, one is a kind of starvation phase, where most of the substrate will be exhausted. Am I right? So, we can assume that S tends to 0, not exactly 0, S tends to 0. Now, if it is tends to 0, I can assume I can solve this problem like this. This is 1 by 0.9, the 0.9 is the mu max value. Then y x by x is 0.5 0.75 and 10 grams per liter is the substrate and this is 0, and x0 is the initial cell concentration is 0.12. So, total time required for the batch process will be coming at 4.32 hours. That means, after if you look at this, this is the how long is the time required to reach the stationary phase, that that we assume is it starts in the this log phase initially and final. This is 4.32 hours that that we can reach this stationary phase. Now, next is that what is the cell mass concentration. Now, if you look at this problem, that what will be the final cell density. What do you mean by density? Density is equal to mass per unit volume. If the fermentation stop after 70% substrate it consumed. So, this problem, that means we shall have to find out when 70% substrate consumed, how much was the initial substrate, 10 grams per liter. How much substrate will be remaining with us? This is 30%. Am I right? So, you multiply it by 0.3, that means 3 grams per liter. Now, so we have the previous equation. I can put the value of S and S0 and S in this equation and find out what is the time required to consume the 70% substrate. It is coming 3.93 hours. Now, initial substrate that cell mass how much there? 12 gram per 100 milliliter. So, this if you divide by that, you will get the 12 that is, this will be how much? You see 12 gram per 100 grams. Am I right? So, this will be about 0.12 gram per 100 milliliter. So, per liter it will be 1.2 gram per liter. So, it has little bit error is there, you please correct it. This will be 1.2 gram per liter. Am I right? So, then we know that dx we have already seen that ln x0 by x, we can write this is x by x0, this will be equal to what? We have seen this is equal to mu max into Tb. Am I right? The time of the batch process. So, I can write x equal to what? x0 into e to the power mu max into Tb. That how much time required to 70%. This is exactly done, only this value will be changed and this value will be little bit altered. That will later on we will correct this. Now, next problem deals with the fed batch reactor. I told you the fed batch reactor is largely used for substrate inhibition, and let us see how we can analyze this process. It is a very interesting problem. Now, if you look at that Nicotiana, this is tabacum, these cells are cultured in a high density production of polysaccharide gum. A start tank reactor was used containing initially 100 liter of media. The maximum specific growth rate of the culture is 0.19 day inverse and yield of the biomass is 0.5 gram per gram of substrate. Now, concentration of the growth limiting substrate is 3% weight by volume. Now, 3% weight by volume means what? Weight by volume means what? This I can write 3 gram per 100 milliliter. Am I right? So, this will be 30 gram per liter. So, this is how we can write. The reactor is inoculated with 1.5 grams per liter of cells and operated in batch until the substrate is virtually exhausted, and media flow then started at a rate 4 liters per hour. Fed batch operation occurs until quasi steady state condition under the quasi steady state conditions. Estimate the batch culture time and final biomass concentration. Second is the fed batch operation is carried out for 40 days, and what is the final mass of the cells of the reactor. Then fermenter is available for 275 days per year with a downtime between runs is about each run between the runs is 24 hours. That means after one run is take about 40 days. Am I right? So after 40 days, that you have 24 hours is the downtime, and how much biomass is produced annually. So this we have to calculate. So I consider this a very interesting problem, and let me explain this problem actually what they are telling. So this is you know that in a fed batch reactor, you take the initial culture, this is the batch mode. So you take the media, take the cell here, and let this organism grow in the batch mode, and you operate the system until unless that whole substrate is exhausted. Then you continuously feed the substrate, and slowly this media is rises, and you keep the substrate concentration, you add in a manner so that this substrate concentration remain much below the inhibition level. This is what is the purpose of this fed batch operation. So we shall have to find out how at the end how much cell mass is produced. Let us see how we can solve it. Now here I told you, whenever we try to solve any kind of problem, first we shall have to write down what are the parameters are given in this problem. Now here what is the parameters is given, mu max value is given, S0 value is given, then x0 value is given, and y x by s value is given. Now from the previous problem we already found that T batch equal to 1 by mu, that is we consider as the mu max, equal to y x by s, s0 minus s plus x0 by x0. You can put these values here that we consider all substrate is exhausted. So s we can consider as a 0. Then we find out that how much exact time is required for this batch process. We can find out that will come around 13.32 days. Now here question comes, what is the final cell concentration, that we have already derived this equation in the previous problem, and we can find out that this is 16.5 grams per liter will be the final cell mass concentration. Now next is that, what I told you, I hope you can remember that this initially we operated in a batch mode. This is the batch mode. Now that this reactor, that we are now continuously feeding in the reactor. So this is the strategy that we have. The cell mass at the start of the fed batch operation is the product of the final batch of the cell concentration. X0 is the X batch into volume is about this. So we know this is the concentration. Concentration is the mass per unit volume, and this is the liter. So this is gram per liter, and this is 100 liter. So if you multiply that, we can find out what is the initial amount of cell present in the batch final after the ending of the batch process. Now we in the fed batch process we have already developed this equation. What is xt? Xt is the final cell mass concentration. Xt equal to x0, f is the flow rate, y x by s is the yield coefficient, S0 is the initial substrate concentration, and T is the time. So so I can write that what is F? F is the volumetric flow rate, volume per unit time. Am I right? Volume per unit time. And what is the value of S0? S0 is the mass per unit volume. So this volume will cancel, this mass per unit time. Now again this is mass of what mass of substrate, this substrate, and then y x by s is y x by s equal to what, the mass of cell by mass of substrate. So the substrate will cancel. The mass of cell. Now per unit time. So if you multiply it by total time, then we will get the what is the final substrate the cell mass concentration. This is how this equation has come. Now simple we put this value, we can find out at the end of the fermentation we are getting 4.05 kg of Now, let us come to the second part or third part of the problem. What is the third part of the problem? That the fermenter is available. Now, the fed batch operation is carried out for 40 days. So, what is the final cell mass? So, that we have find out that T we consider 40 days, we find out. And third part is that fermenter is available 275 days per year, with a downtime between the runs is 24 hours. How much biomass is produced annually? So, let us see how you can solve this. This we what you can the run that per run, how much is the cell mass we can produce. This is the total cell mass, this is the initial cell mass. So, what is the per run, how much this is the final. We previously we find out how much is the cell mass concentration. We can find out it is 4.05 kg, that is mean 4050 gram, and this is the initially cell mass that was present. So, if you multiply that, you will get that how much cell mass you produce per run. Now, how many runs you can conduct? This is the batch time, that this is how much 13.432, and then fed batch time how much is required 40 days, and downtime is required 1 day. The total time is required like this. So, how much run, because we shall have to operate 275 days. So, divided by 54.32, that means 5.06 run you have to conduct. Am I right? If one run we can produce 3.9 kg of cell mass, the 5.06 run, how much is cell mass is produced? 19.73 kg. So, we can easily find out that total amount of cell mass is produced. Now, last problem that we have, lactobacillus casei. This is also very interesting, but similar to feed batch process. Lactobacillus casei is propagated under the essentially anaerobic condition to provide the starter culture for manufacturing switches. Now, the culture produce lactic acid as byproducts for energy metabolism. The system has the following characteristics. These are the different values are given. The start tank reactor is operated in fed batch mode at quasi steady state with a feed flow is 4 cubic meter per hour, and feed substrate concentration is 80 grams per cubic meter, kg per cubic meter. After 6 hours, the liquid volume is 40 cubic meter. What is the initial volume of the culture? That you have to find out. What is the concentration of the substrate at the quasi steady state? What is the cell mass concentration at the quasi steady state? How much biomass is produced after 6 hour fed batch operation? So, I am quickly go through this because so you see that we have seen that V equal to V0 plus Ft. Now, V0 we know the initial volume we shall have to find out the V0 V minus Ft. So, V is given, this is 40 liters or 40 cubic meter, then and F is 4 meter cube per hour, and this is the 6 hours time. So, we can easily find out the initial volume. Initial volume is very easy, you can determine that. Now, in the next part, that we know that you can find out the dilution rate. Dilution is nothing but flow rate by volume. This is 0.1 hour inverse, mu equal to D under quasi steady state condition. This is, and then S value is this. We can put the different values in this equation, then we can find out the steady state substrate concentration. Now, once you know that, the next stage is that we shall have to do the substrate balance across the reactor. Equation is rate of input plus rate of generation, rate of output, rate of consumption plus the accumulation. So, we can write whole equation like this, and this is the final equation we will be having. And if we consider QP equal to, here we do not consider other than cell mass as a product. So, we can assume QP the product specific rate of product formation equal to 0, then our final equation will be this. This equation, if you further analyze, you will get that this equation, and then as steady state condition, that rate of accumulation of the substrate, that should be equal to 0. That should be equal to 0. So, this is exactly what is written there, and mu equal to d, that is then we can write this equation in this form. This is x equal to d S0 d by y x by s by ms. Ms is the maintenance coefficient, and if you put the value you can get the final cell mass concentration. And then the total biomass will be what? Total biomass into x x into x is the cell mass concentration in the final volume, and x is the cell mass concentration in the initial volume. So, we can find out this for this value, final cell mass concentration, the amount is 561.6 kg, and the initial will be this. So, the difference will give you the total biomass that is produced in the system. You know that so this particular lecture I try to cover several problems, 4 different problems, and I hope the batch and fed batch process you can analyze very nicely, and you can solve the problem with respect to substrate conversion, with respect to cell mass formation, that we can easily monitor. Thank you very much.